Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024, which explores the intricate world of the immune system in dogs suffering from a common and challenging condition: chronic inflammatory enteropathy, often referred to as CIE. Chronic inflammatory enteropathy encompasses a group of disorders where dogs experience persistent and recurring signs of gastrointestinal disease. This condition is a major cause of ongoing vomiting and diarrhea in our canine companions. While the exact causes remain unknown, it's understood that a complex interplay of immunological, dietary, genetic, and environmental factors likely contributes to its development. However, a key aspect is the role of the immune system itself. Chronic inflammatory enteropathy is largely considered an immune-mediated disease, where an overactive and abnormal immune response to various antigens within the gut leads to the troublesome clinical signs we observe. Within the complex machinery of the immune system, T cells, a type of white blood cell, and the substances they produce are thought to be particularly influential in the development of intestinal inflammation. In humans with inflammatory bowel disease, a condition with similarities to chronic inflammatory enteropathy in dogs, T cells located in the lining of the intestine and the layer beneath it, known as the lamina propria, appear to have a significant impact on the immune response. Interestingly, inflammatory bowel disease in humans is also recognized as a systemic illness, affecting other parts of the body in many patients. Some researchers believe this occurs due to the migration of immune cells from the gut to other locations. While extraintestinal manifestations are not as commonly reported in dogs with chronic inflammatory enteropathy, changes in the immune cell populations in their peripheral blood have been observed. Diagnosing chronic inflammatory enteropathy in dogs involves a thorough process of ruling out other potential causes of intestinal inflammation. This is often followed by examining tissue samples under a microscope to look for evidence of inflammatory cells within the intestinal lining. Because the immune system plays such a central role, treatment often involves medications that suppress or modify the immune response. This is why the condition is sometimes referred to as steroid-responsive enteropathy or immunosuppressant-responsive enteropathy. Advances in our understanding of the immune mechanisms behind inflammatory bowel disease in humans have led to the development of effective immunotherapies and the identification of blood-based markers that can help predict how patients will respond to treatment and how their disease might progress. However, our knowledge of the specific immune changes that occur in dogs with chronic inflammatory enteropathy is still somewhat limited. Therefore, the aim of this particular study was to investigate the alterations in immune cells, specifically T lymphocytes, in both the bloodstream and the intestinal tissues of dogs diagnosed with chronic inflammatory enteropathy. The researchers had several specific objectives. First, to describe the main populations of T lymphocytes in the peripheral blood, within the intestinal lining, and in the lamina propria of dogs with chronic inflammatory enteropathy using a technique called flow cytometry. Second, they aimed to compare these T lymphocyte populations between dogs with chronic inflammatory enteropathy and a group of healthy dogs. Finally, the study sought to determine if there was any association between the T lymphocyte populations in the blood and certain inflammatory markers derived from routine blood tests, as well as the clinical signs, endoscopic findings, and microscopic tissue changes observed in dogs with chronic inflammatory enteropathy, in the hope of identifying potential biomarkers for the disease. The study involved a total of 42 dogs, 26 diagnosed with chronic inflammatory enteropathy, and 16 healthy dogs serving as a control group. The researchers carefully documented various characteristics of the dogs, including their age, sex, breed, body weight, and body condition score. Importantly, there were no significant differences in these basic characteristics between the two groups. To evaluate the health status of the dogs, complete blood counts were performed, and in the dogs with chronic inflammatory enteropathy, the severity of their gastrointestinal signs was assessed using established clinical scoring systems. During the diagnostic process for the dogs with chronic inflammatory enteropathy, they underwent upper gastrointestinal endoscopy, a procedure where a small camera is used to visualize the inside of the digestive tract, and tissue biopsies were collected for microscopic examination. The healthy control dogs also underwent upper gastrointestinal endoscopy for the purpose of sample collection, as they were anesthetized for other routine procedures. The appearance of the gastrointestinal tract during endoscopy was scored by experienced veterinarians, and the microscopic changes in the tissue biopsies were evaluated by a pathologist using standardized scoring systems. For a detailed look at the immune cells, additional tissue biopsies were collected from the small intestine of all dogs and processed to isolate different populations of lymphocytes. Those within the intestinal lining, known as intraepithelial lymphocytes, and those in the lamina propria. Blood samples were also collected to isolate lymphocytes from the peripheral blood. These isolated lymphocytes were then analyzed using flow cytometry, a technique that allows researchers to identify and count different types of cells based on the proteins they express on their surface and inside them. The researchers focused on various subsets of T lymphocytes, including helper T cells, cytotoxic T cells, activated T cells, regulatory T cells, and T cells that produce specific immune signaling molecules like interferon-gamma and interleukin-4. The results of the study revealed several significant differences in the T lymphocyte populations between the healthy dogs and those with chronic inflammatory enteropathy. In the peripheral blood, dogs with chronic inflammatory enteropathy had higher numbers of activated helper T lymphocytes and regulatory T cells compared to the healthy dogs. While most of the lymphocytes in the peripheral blood of both groups were T cells, with helper T cells being more numerous than cytotoxic T cells, the chronic inflammatory enteropathy group showed a notable increase in these specific activated helper T cells. Regulatory T cells, another important subset involved in controlling immune responses, were also found to be significantly elevated in both the percentage and absolute numbers in the peripheral blood of dogs with chronic inflammatory enteropathy. When the researchers examined the T lymphocytes within the intestinal lining, they observed a different pattern. In the intestinal epithelium of dogs with chronic inflammatory enteropathy, the percentage of helper T cells and activated helper T cells was lower compared to the healthy dogs. Conversely, a higher percentage of activated cytotoxic T cells and T lymphocytes producing interferon-gamma were found within the intestinal lining of the dogs with chronic inflammatory enteropathy. This shift resulted in a significantly lower ratio of helper T cells to cytotoxic T cells in the intestinal epithelium of the diseased dogs. In the lamina propria, the layer beneath the intestinal lining, the study found a higher percentage of regulatory T lymphocytes in dogs with chronic inflammatory enteropathy compared to the healthy control group. Interestingly, when comparing the three different immune compartments, peripheral blood, intestinal lining, and lamina propria, the highest percentage of regulatory T lymphocytes was found in the lamina propria. The researchers also investigated potential associations between the T lymphocyte populations in the peripheral blood and various clinical and pathological findings in the dogs with chronic inflammatory enteropathy. They found a moderate negative correlation between the severity of microscopic lesions in the duodenum, a part of the small intestine, and the percentage of interferon-gamma producing T lymphocytes in the peripheral blood. This suggests that as the intestinal inflammation worsened, the proportion of these specific T cells in the bloodstream tended to be lower. Furthermore, the study evaluated several inflammatory markers that can be easily calculated from a routine complete blood count, such as the neutrophil to lymphocyte ratio, the platelet to lymphocyte ratio, and the systemic immune inflammation index. They observed a moderate positive association between the systemic immune inflammation index and the severity of microscopic lesions in the duodenum as assessed by two different scoring systems. This indicates that a higher overall measure of systemic inflammation as reflected by this index tended to correlate with more severe inflammation in the small intestine. The dogs with chronic inflammatory enteropathy also showed significantly higher platelet counts and systemic immune inflammation index values compared to the healthy dogs. The findings of this study provide valuable insights into the complex immune alterations that occur in dogs with chronic inflammatory enteropathy. The observation of increased activated helper T cells and regulatory T cells in the peripheral blood suggests a state of systemic immune activation in these dogs. The increased regulatory T cells might represent an attempt by the body to counteract the excessive inflammation, although their precise role in this disease still needs further investigation. The changes observed within the intestinal tissues paint a picture of a more localized and potentially aggressive immune response. The lower numbers of helper T cells and the higher numbers of activated cytotoxic T cells in the intestinal lining, along with increased interferon-gamma production, are consistent with a pro-inflammatory environment at the site of the disease. The decreased ratio of helper to cytotoxic T cells in the intestinal epithelium is a finding commonly reported in both human inflammatory bowel disease and chronic inflammatory enteropathy in dogs. It's hypothesized that an imbalance in the regulation of cytotoxic T cells in the intestinal lining may contribute to the ongoing inflammation and damage to the intestinal barrier. The increased interferon-gamma production further supports the idea of a type 1 immune response being dominant in the small intestine of these dogs. The correlation observed between the peripheral blood interferon-gamma producing T lymphocytes and the severity of duodenal lesions is intriguing. It might suggest a dynamic relationship where these cells are recruited to the inflamed intestine, leading to a decrease in their numbers in the bloodstream, although more research is needed to confirm this. Similarly, the association between the systemic immune inflammation index and the severity of duodenal lesions indicates that the overall level of systemic inflammation, as measured by this index, reflects the degree of tissue damage in the intestine. This suggests that the systemic immune inflammation index could potentially serve as a biomarker to assess disease severity and monitor response to treatment in dogs with chronic inflammatory enteropathy. In conclusion, this study highlights significant alterations in both the peripheral and intestinal T lymphocyte populations in dogs suffering from chronic inflammatory enteropathy. These findings underscore the important role of the adaptive immune response, driven by specific subsets of T lymphocytes, in the development and progression of this disease. The study also demonstrates the utility of flow cytometry in characterizing these immune cell populations in different compartments of the body, offering advantages over other techniques in veterinary medicine. The researchers suggest that further investigation into the changes in lymphocyte populations and their products is crucial for a better understanding of chronic inflammatory enteropathy in dogs, and for the development of improved diagnostic and therapeutic strategies. The potential of peripheral interferon-gamma producing T lymphocytes and the systemic immune inflammation index as immunologic and inflammatory markers for this disease warrants further research with larger groups of animals to determine their reliability and clinical usefulness. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.